The moderator for this evening's panel is Dr. David Kessler, the Dean of the UCSF School of Medicine. Before coming to UCSF 2 years ago, Dr. Kessler had been Dean of the School of Medicine at Yale for 6 years, and from 1990 to 1997, he was Commissioner of the U.S. Food and Drug Administration. During his tenure at FDA, he made quite a mark by streamlining the approval process for important drugs and medical devices, by working to limit the marketing of tobacco products to children, and to improve the nutrition labels on foods. I read his handiwork on the yogurt packet every morning. Dr. Kessler serves as UCSF's representative on the Independent Citizens Oversight Committee, the board that was established by Proposition 71 to oversee California's stem cell program approved by the voters in the last election. Dr. Kessler, please.
Thank you very much, Dr. Bishop. Yesterday, one of my colleagues said that what is really new about stem cell science is the amount of interest in it. The term embryonic stem cell itself was coined 25 years ago by UCSF's Gail Martin, who co-discovered these cells in mice. The sine qua non of an embryonic stem cell is that it has potential, the potential to become an array of different cell types. What we're going to focus on tonight is what the real potential, not only of stem cells, but the whole field of stem cell research really is. Let me introduce tonight's panelists. We are honored to include in our discussion 3 recipients of the Nobel Prize for Medicine or Physiology. Dr. J. Michael Bishop is the Chancellor of UCSF. His co-discovery that cancer is caused by normal genes gone awry changed our fundamental understanding of cancer research. Dr. Stanley Prusiner is Director of the Institute for Neurodegenerative Diseases. Persevering over 2 decades, Dr. Prusiner opened up a new field of brain research by demonstrating that a protein can cause disease. Our third Nobel laureate, Dr. Arvid Carlsson, is an emeritus professor of pharmacology at the Gothenburg University. His discoveries about cell communication in the nervous system shed crucial light on normal brain function and led to the development of drug therapy for Parkinson's disease. We are also joined by several UCSF scientists on the frontier of stem cell research. Dr. Arnold Kriegstein is a neural stem cell researcher and director of UCSF's Institute for Stem Cell and Tissue Biology. Dr. Renee Rayopera is an embryonic stem cell researcher and co-director of the UCSF Human Embryonic Stem Cell Center. Dr. Elena Gates, interim chair of the Departments of Obstetrics, Gynecology, and Reproductive Sciences, is a leader in addressing and navigating the ethical issues of embryonic stem cell research. Dr. Jeffrey Bluestone, director of the UCSF Diabetes Center, is one of the world's foremost experts in transplantation immunology. I'm going to get the discussion going with our panelists. Later, we'll have time for questions from the audience, both here at Genentech Hall and at our telecast sites at Cole Hall and the Gladstone Institutes. If you have a question, please write it on one of the cards that were handed out as you came in. Ushers will collect your questions. We will post answers to as many questions as possible on our website, stemcellfacts.ucsf.edu. Let me get the discussion going. Dr. Kriegstein, what is a stem cell?
Briefly, stem cell is an undifferentiated cell that, as you indicated in your introductory remarks, has the ability to become any cell type in the body. The other property of stem cells is that they can self-renew, so that when they divide, they can produce multiple copies of themselves. And they can do this indefinitely. And so they are a potential inexhaustible source of cells that could be used for, say, cell replacement therapy.
Dr. Rayopera, I just asked Dr. Kriegstein what is a stem cell. Let me ask you what is an embryonic stem cell, and what's the difference between an adult stem cell and an embryonic stem cell?
Well, I work on human embryonic stem cells, so I'll talk about human embryonic stem cells. These are stem cells that are derived from human embryos that are generally donated in the clinic, in infertility clinics, by patients who are generally infertile and have excess embryos that are either of poor quality or they've finished their childbearing, so they wish to donate their embryos. So human embryonic stem cells are actually derived from human embryos. Okay. The process of deriving the human embryonic stem cells actually begins with the fertilization of an egg with a sperm. The cell, that's the first cell of the embryo. It goes on to divide several times, and by day 3 in human development, we actually have 8 cells. Those cells go on to continue to multiply, and by about days 4 and 5, we have about 100 cells in the embryo, and they're forming a ball of cells. The outer layer of cells would actually normally attach an embryo to a uterus, but in a dish, what happens is the outer cell layer generally degrades, and there's an inner cell mass. Normally, that inner cell mass would form many different types of cells of the body. But in a dish, again, sometimes these cells, they begin to proliferate, and they expand, and they make colonies on a plate. And that's the beginning of an embryonic stem cell line. So it comes from the inner cell mass of an embryo that's like a ball of cells of about 100 cells. Okay. Now, the special thing about human embryonic stem cells compared to adult stem cells is human embryonic stem cells, we have not yet seen a cell type in the adult body that cannot be differentiated from human embryonic stem cells. So their potential is very great. But associated with that potential is a pretty serious backlash, and that's that we can't quite drive the process down just one lane. So it's difficult to get a 100% pure culture of neurons, for example, from a cell type that wants to make every different cell type in the body. So in contrast to human embryonic stem cells, adult stem cells, they actually can be found in the adult body or in mature tissues, and they have more limited potential. So neural stem cells, in general, are going to make cells such as neurons. Hematopoietic stem cells are going to make things like cells of the blood lineage. So their potential isn't as great at all. They're limited to 1 or 2 jobs, basically, but the side effects aren't as great either.
So as I stand before you tonight, in my body, there are stem cells?
Arno can answer that.
I see a pattern developing tonight. Well, there's certainly stem cells in certain organs of the body. We're all familiar with the way the skin can heal itself. The bone marrow contains stem cells. The liver can regenerate in many cases as well. And interestingly enough, the brain, which for many, many years had been thought to be a site that doesn't sustain any new cell birth, is actually a site of neurogenesis in the adult. So new nerve cells are born in our brains here in the audience as adults. And the origin of those cells are neural stem cells that are resident in the adult brain.
So we now know, again, which organs are their stem cells?
Well, there are still stem cells to be discovered. There's the hope, for example, that there'll be a stem cell in the adult pancreas, and that's one of those areas that people are still looking for.
So let me just clarify this. In the adult stem cell, let's say in the pancreas or in the liver or in the brain, that adult stem cell can become what?
Well, as Renee suggested, those adult stem cells are different than the embryonic ones. They have a much more limited potential. So the nerve stem cell, neural stem cell, has the ability to make the kind of cells that are found in the nervous system. Nerve cells, glial cells, oligodendrocytes, a variety of different cell types. Because a single cell can make all of those cell types, it has multipotentiality, but it doesn't have pluripotentiality. It can't
just be careful on those terms. Which is greater, multi or pluro?
Pluri is greater. So let me just run through the definitions. Totipotential is the ability to make every cell. And so the fertilized egg, after fertilization, has that ability. It makes not just the embryo, but it also makes the placenta. So it makes all the cell types in the embryo. As you proceed along the developmental pathway, the potential becomes progressively restricted. And so after totipotentiality, you reach a pluripotential state, which allows the cells to make most but not all the tissues. With further development in certain organs, those stem cells become further restricted, and they might be multipotential, which means they can make multiple cell types, but not all cell types. Further down the line, they could be tri- or bipotential, which means they could make 3 or 2 cell types. And then ultimately, you wind up with a differentiated cell that can't divide anymore, and that's the end product.
Can you go backwards in the process?
Well, that's a very interesting question. There have been some reports of that kind of de-differentiation occurring, but they're still quite controversial, and it's an area that really needs to be explored.
So let me understand. You work on human embryonic stem cells.
Yes.
Dr. Quickstein, you work on?
Fetal neural stem cells, mostly.
Now just explain the difference.
My cells have more potential. Well, that is simply the difference.
So when we use the term, even using the term embryonic stem cell, doesn't necessarily mean the same thing.
Well, I almost always define embryonic stem cells as stem cells derived from a human embryo. That's a fact. And most commonly, that's from a blastocyst in the inner cell mass, as I explained.
Very early on in the embryo.
That's most commonly. There are also embryonic germ cells and some other cell types that are not quite as useful because of some genetic problems.
Now, you or your colleagues work on what kind of cells?
Well, let me just emphasize a little bit before I answer that question, the fact that when you talk about embryonic stem cells these days, mostly what is referred to is the blastocyst that Renee described so well, that inner cell mass surrounded by a shell. And that's after 3 to 5 days of fertilization. And in the human body, this is before the embryo implants in the uterus. It's a pre-implantation embryo. But the term embryo can be applied to anything from the point of fertilization all the way down to a fetus, which has organs and limbs and looks like an identifiable individual. And so the term, because it's not precise, can cover a whole range of different stages in development. But it's that early multi-stage blastocyst that people really derive stem cell lines from. The cells that I've been studying occur much later, once the organs have already started to differentiate and you can see an identifiable brain. And then neurons, the nerve cells in the brain, are developed. They divide from precursor cells. Those precursors come from neural stem cells, and those are the cells that I'm studying, the cells that actually give rise to the various cell types in the developing brain.
And some of our colleagues at UCSF study cells later on in the process?
Absolutely. So as I've mentioned, there are resident stem cells in the adult brain. And Arturo Alvarez-Bulla, who's a colleague of mine here at UCSF, was the first to really demonstrate that there are areas around the ventricles, deep inside the brain, where these kinds of cells continue to be born from stem cells in the adult. And the nature of those stem cells turns out to be very similar to the fetal or embryonic stem cells in the nervous system that I've been studying. And so there's a continuum of stemness from the early embryo all the way to the adult brain, where neurons continue to be produced.
Dr. Bishop, let me step back for a moment. Bigger picture. How do scientific fields develop, and what should we expect, and where should we focus our attention from stem cell research?
Albert St. Georgi, who won the Nobel Prize for his work on vitamin, once wrote an essay on this issue of how science progresses. He said that when scientists write about their work and their discoveries in retrospect, they describe a perfect straight line, relentless progress from one point to another. The reality, he said, looks like the track of a drunken sailor. Many of the steps backwards in the wrong direction by that drunken sailor represent either erroneous hypotheses or failed experiments. And furthermore, he said, many of the steps in the right direction represent the totally unexpected. So what should we expect? The unexpected. And stem cell research demonstrates that cloning the sheep dolly was, in a sense, the ultimate stem cell experiment. The outcome was utterly unexpected by the overwhelming majority of scientists because as long as science has been studying the development from the fertilized egg to the mature organism, it has been assumed that once a mature tissue has been achieved, the cells in that tissue are frozen in place. They can't change the program anymore. They can't go backwards. They can't go to another form of tissue. But what the cloning of Dolly demonstrated for the first time is that a mature cell can indeed be reprogrammed all the way back to the state of the embryonic stem cell. This was absolutely a stunning and unexpected discovery, and I'm sure that since it's very early days in stem cell research, we're going to see a lot more such utterly unexpected and unpredictable results. One wise man once said scientists should never make predictions, so I'm not going to predict very much. But I think some reasonable expectations in the near term, where we're going to see improvements in the efficiency with which embryonic stem cell lines can be isolated. We're going to see perfection of our ability to grow them strictly with human materials so they're not contaminated by animal materials. We're going to see improvements of our ability to do the kind of experiment that led to the cloning of Dolly. Right now, those experiments are, they don't work very often. We're going to see fundamental insights into the chemical signals that tell stem cells to go one way or another to become a pancreas cell or a blood cell or whatever. And I think we're going to see a much better definition of adult stem cells. As you heard, there's controversy about these cells, whether all organs possess them or not. We could only name 3 or 4 where we're pretty certain that there are adult stem cells present. And where should we focus? I think it's generally agreed that we ought to be focusing on the isolation and characterization of more lines, because as we study the existing lines, we're finding imperfections in them, we're finding problems as they propagate. We need to know a lot more about what happens when we propagate those lines. We need to seek ways to signal where a stem cell should go, what type of tissue it should become. This is fundamental to clinical application of stem cells. And we need to settle the debate over the plasticity of adult stem cells. There is a big controversy with most people believing that adult stem cells have little plasticity. They really can't be used to form different kinds of tissues. They can only be used to form the tissue from which they are derived. But there is a minority who believe that adult stem cells can, under certain circumstances, be persuaded to adopt a different identity, to, instead of being a blood stem cell, to become a heart stem cell. And the significance of that, were it true, is that it might take some of the pressure, political and ethical pressure, off the use of embryonic stem cells. So we really need to settle that issue. And I think it's an issue that's accessible to study now. So I would expect that issue will be settled in the next 5 or 10 years.
Dr. Prusner, your work has opened up a new window on neurodegenerative diseases. Where do you see a role for stem cell research in the study of these diseases, diseases such as ALS, Huntington's, and Alzheimer's?
Well, I think we're very excited about all of this because if you step back and look at the field of neurodegenerative diseases, which includes Alzheimer's disease that affects 5 million people in the United States, Parkinson's disease that you'll hear about from Dr. Carlsson in a moment, 1 million people, ALS, or called Lou Gehrig's disease, the prion diseases such as mad cow disease, Huntington's disease that Dr. Kessler just mentioned, a degenerative inherited brain disease, and even multiple sclerosis. What you see is that we've had one and only one really effective drug in the last 40 years. So we see stem cells as a new way of approaching these horrifying diseases that devastate the lives of so many people. What is that one drug 40 years ago? L-DOPA. And L-DOPA, while it ameliorates the symptoms of Parkinson's disease, it doesn't stop the underlying process. Now, that isn't to say there aren't a lot of drugs in the pipeline and that there are new drugs coming all the time and that some of these will be effective, but no drug right now has achieved the acclaim of L-DOPA, where we know that it really does something and over a prolonged period of time. So in the field of neurodegenerative diseases, we're looking for new answers, and we see stem cells as a way to do this. Now, I think you can divide these diseases into two groups, and you'll hear more about Parkinson's disease in a moment, but that's group one, along with ALS and multiple sclerosis. In these diseases, we need to replace essentially one cell type in a very localized area. And it seems to me that stem cells may actually be a therapeutic in the nervous system. In Alzheimer's disease, Huntington's disease, and the prion diseases, I think it's going to be harder. I think it's going to be much harder because there the degenerating cells are found throughout the brain. So there are many different types of neurons that we will have to replace. But let me give you an example of how I think this can work and what we're looking to do. For instance, in Alzheimer's disease, we don't think that in our lifetime, in the lifetimes even of our children, that we're going to cure Alzheimer's disease with stem cells. Now, maybe that's a little pessimistic, but what I think we can do is we can create models in mice with human embryonic stem cells or human adult stem cells, depending, as Mike Bishop said, on how this plays out, where we'll be able to have a perfect model. What's a perfect model? Why is that so important? Because it allows us to test drugs or compounds that lead to drugs very early. Right now, we don't have a perfect animal model of Alzheimer's disease. Same thing with Huntington's disease. While we have great models of prion diseases, what we need, for instance, to test drugs is we need to be producing human prions and human cells inside the brain of a mouse. So all of this, I think, is going to be possible with stem cells, and I've just touched the surface. So I think the route to the future is very exciting.
Dr. Carlsson, some scientists say that Parkinson's is one of those diseases that is likely to be among the first treated with stem cell therapies. Your thoughts, sir?
I think here I have to play the role of the doubting Thomas. I think we should go back actually 100 years and go back to one of the great pioneers, Ramon Cajal. He said that when a nerve cell in the adult brain is lost, it cannot be replaced. And the data that we have now are not from all parts of the brain, but one major part of the brain, of course, is the cerebral cortex. And another, for that matter, is cerebellum. And regarding those, there is now good evidence, and from using different techniques, that tend to confirm what Cajal stated. There are different approaches insofar as the cerebral cortex are concerned that show that those nerve cells that we have in our cerebral cortex are the ones that were formed before we were born, or possibly during the first couple of years after we were born. After that, no more nerve cells are formed in the cerebral cortex of the human brain. And the same thing, there are data for the cerebellum, as I said. So in other words, what it means is that if a nerve cell is lost in these parts of the brain, it cannot be replaced because there is no formation of new nerve cells in these parts of the brain. So what, of course, we don't know about other parts of the brain yet, but there are very good techniques now to study these things. So I would guess within maybe 5 years or so, we will have much more information about this. And I feel a bit pessimistic. I feel that what has been found concerning the cerebral cortex and the cerebellum will be true of many other parts of the brain. That perhaps there are some Perhaps the hippocampus could be an exception because there are certain data in favor of that. But for most parts of the brain, I don't think there is much hope. And if we then come to Parkinson's disease, here we have a loss of the dopamine-forming nerve cells in the lower brainstem. And the evidence that we have now is not encouraging. I mean, there are animal data. There were some suggesting that perhaps in the rat there could be some new formation of dopamine-forming cells from stem cells, but that could not be confirmed by others. And also, if you go to evidence of transplantation, of course, there are no data yet on stem cells, but we do have very good data, I think, from fetal dopamine cells being transplanted into the brains of Parkinson patients. There are two good studies, very well-controlled studies from the US, and they showed that there is no therapeutic, no improvement of the symptoms of Parkinsonism, but rather there were some disturbing side effects in the form of involuntary movements. So I feel pessimistic about Parkinson's disease as one example. I feel pessimistic about Alzheimer's, for example, also about perhaps some other disorders.
Arnold, comments? Our guest from Gothenburg University, your thoughts?
Well, absolutely. I certainly would not contradict anything that Dr. Carlsson said. In fact, I agree completely, but I think it's a difference of viewpoint. It's an example of whether the glass is half full or half empty. Because you're absolutely right. For 100 years, the dogma has been that you're born with a certain number of nerve cells, and from then on, you start losing them. So the surprising thing is that this discovery that there are neural stem cells that in the adult brain violate the dogma. They actually continue to produce nerve cells. And it is true that we're talking about just two restricted areas of the brain. One is the periventricular region, deep inside the brain. And the nerve...
Where is that for the rest of us? I mean,
What does it do?
Sure. Well,
Point to it.
it's very deep. In the core of the brain, there are spaces, fluid-filled spaces known as ventricles. And lining the walls of those fluid-filled spaces is a thin layer of cells that actually continue to divide and produce nerve cells. And the nerve cells they produce, even though these are produced deep inside the brain, are not usually destined to go to the brain directly, but rather to the olfactory bulb. So they have a long distance to travel, and they migrate from where they're... Yes, the olfactory bulb is involved in olfaction for smell. It sits behind the nose. And so these nerve cells collect and they travel in streams and they go to the nose and they continuously turn over. They replace cells that die. And so you don't have a net increase in the size of your olfactory bulb, but there is a continuous replenishment of these cells. And so that's a very surprising discovery, that nerve cells of any kind are generated throughout life. And the second area of the brain where this happens is even more surprising. It's an area very related to the cerebral cortex, which is the hippocampus. It's a more primitive form of cerebral cortex. And in that area as well, in one area known as the dentate gyrus, there are daily produced new nerve cells. And some of those die, but others survive. And so there is also in that organ a turnover of nerve cells throughout life. And what's surprising is that the hippocampus is an organ of learning and memory. It might be the last place you'd expect new cells to continuously form, but oddly enough, that's one of those few areas where that happens in the adult. And it's unclear what those cells are for, whether they're involved in learning or memory, whether they're involved in mood or affect. They may even be the basis of depression when they're absent. There's some studies suggesting they're related to depression itself. So there's a lot we still need to learn about all this. But because this has changed our dogmatic view that the brain is static, I think it really is a very hopeful view of what's going on.
likely we're going to find these stem cells in other areas of the brain other than the two you've just identified?
In normal, healthy individuals, I doubt it, because as Dr. Carlsson mentioned, this has been looked at for 100 years now, and with modern techniques, it's been looked at again. There was a report a number of years ago that the cerebral cortex could sustain normally other areas of neurogenesis, but that's now been pretty widely discredited. So I think that there probably are no new discoveries to be made about new nerve cells being born in most parts of the brain, in healthy brain. And I mention that because following brain injury in stroke and traumatic lesions, there are lots of papers now suggesting that there can be neurogenesis. To some of these areas I'd mentioned earlier, around the ventricles, that normally produce cells destined for the olfactory bulb, following certain kinds of strokes, those cells appear to diverge toward the area of injury. Now, whether any of them become nerve cells or whether they become other kinds of cells, like glial cells, which are a different kind of cell in the brain, is still being discovered and described. But it appears that following a pathological lesion, things can change. And I'd like to, just for a brief mention, describe lower vertebrates, not humans now, but reptiles in particular, which have a very primitive cortex that continues to grow throughout life. There are new cells that are added to the cerebral cortex of these animals throughout life, and following insults and injury, they can regenerate parts of the cortex the way most of you probably know they can regenerate a tail. And we don't have that ability in our brain. But the fact that these cells in the adult brain can integrate, can form circuits, can develop projections that go to the right targets in the territory of the fully developed adult brain is remarkable. And so is another example of looking at the glass either half full or half empty. I find that this is a whole new area of discovery now where anything can happen, where we might be able to figure out how to tweak the cells that normally make olfactory bulb neurons and get them to make other kinds of cells, maybe replace cells that are lost following some of these degenerative disorders. So I think it's a very hopeful time right now. And of course, it's at an early stage, so we can't really describe the full potential of these cells. But it's an area that I think is going to be an area to watch over the next 5 to 10 years.
Stan?
Yeah, I'd just like to say that I think we're talking about two subjects here. One subject is regeneration in the brain with the endogenous stem cells of the brain. The other area we're talking about is learning to tweak transplanted cells, as you mentioned, where we take embryonic stem cells or adult stem cells, we program them to become dopaminergic neurons, the neurons involved in that die off in Parkinson's disease. So now we implant those neurons into the brain. The problem will be to make the connections, but that's a part of the challenge of these studies, is to learn how to modulate, trigger, signal, send the right messages to those cells. And what I was trying to say before was, I don't think in Alzheimer's disease, and so we agree about the pessimism there of doing that anytime soon, but where we're talking about one cell type and a limited area of the brain, I think we have a better chance.
Dr. Carlsson.
It's interesting to bring in the lower animals. And actually, if you go further down to the salamander, it's even smarter in making new, if you lose a little bit of the brain. Then if you go to the reptiles, and as you go up then to the rodents, for example, and primates, non-human primates, and then to humans, you can see very clearly that as you move upwards, the ability to generate new neurons goes down very dramatically. So in the human brain, that is the minimum. And then the question comes, what is could be the explanation for this? Why have we lost our ability, so much of our ability to form new neurons? And there is a very interesting theory here that I really believe in strongly. It's simply that the circuitry that was formed at an early stage and then later finally established itself, circuitry of different neurons in very complex circuitries, they simply do not accept any newcomers. And why don't they accept any newcomers? That is because in the human brain, which is in contrast to the salamander, memory is so enormously important. And the memory resides in those neurons. So if you lose one neuron and you replace it with another one, if you could do it, that wouldn't be of any help. Actually, it could be quite disturbing to bring in naive neurons who didn't have the wisdom of the one that got lost, and therefore you can have actually problems with your memory if you accept newcomers. So that is probably what has happened with the human brain. We have had to reject any newcomers in order to preserve our fantastic ability to store information from very early life. Throughout life, we are storing information. We cannot accept newcomers. That is an interesting theory that I feel I am fascinated by.
So Dr. Bishop identified early on a very important question that needs to be settled, that he thinks, in the next 5 years, the ability of adult stem cells and what's the real potential of them. As I hear it, here is another very important question that needs to be settled over the next, can it be settled in the next 5, 10 years, do you think? Whether, in fact, you can, the brain can accept newcomers?
Yes, I simply, by using techniques that are available now, and there are some really new techniques where you actually start out from measuring the the radioactive carbon that we have in the air, and how that is equilibrated with newly formed cells. On the basis of that, and based on the fact that the atom bombs cause an increase in the radioactivity, you can actually tell the age of cells by looking at the radioactivity. So therefore, and that is one of the methods that have been used that end up with the conclusion that in the cerebral cortex and in the cerebellum, the neurons that we have are as old as we are. And of course, this technology can be used for just any part of the brain, so it's just to go on and do all this, and that can of course include also brains of disease of patients with who have had disorders, and then we can see whether what you suggested, that if you have some disorder such as stroke, there is then formation of new cells. Well, that all this can be done, and I think it can be. It will probably be done within the next 5 years or so.
What's the advantage or disadvantage, I mean, of a stem cell, if this could be done? If this could be done in the area of the brain where there's dopamine depletion and Parkinson's develop, what's the advantage between stem cell and a drug like L-Dopa? What's the advantages and disadvantages?
I see no advantage with these stem cells because they are not likely to turn out to be therapeutically useful. So we are left with L-Dopa so far. But rather, but I think what the direction of the research should go in, it should go in a very different direction in prevention. I think this is a very interesting area in the research into Parkinson's disease, because we do have indications that there are molecules in our environment that could be very important for the etiology of Parkinson's disease. And to go further into that, I think would be much more fruitful than to pursue this idea of stem cells and transplants and all that.
Perhaps I could just make one relatively brief comment, is that I certainly agree with Dr. Carlsson that replacing some neurons in the brain, especially in the cerebral cortex, is going to be daunting, if not totally impossible. On the other hand, one doesn't only use stem cells as a vehicle for replacing cells that are missing. You can use them as delivery vehicles to bring growth factors or genes to areas where they may be needed. So, for example, the dopaminergic cells that are dying in the area of the midbrain in Parkinson's disease might be preserved if growth factors could be delivered to the environment of those cells. And there's one in particular called glial cell-derived neurotrophic factor, or GDNF, which has been shown to keep these cells alive and actually counteract some of the effects of cell death in models of Parkinson's disease. And so you can engineer a stem cell to express this GDNF gene to produce the growth factor. And the stem cell has, some of them have an intrinsic ability to migrate and integrate into certain areas of the brain. You could then deliver these stem cells, this is theoretical at this point, into the midbrain. They could surround the areas of the dopamine cells that are dying, and by secreting and releasing this growth factor can prevent them from dying. So that would be the use of a stem cell as a delivery vehicle, not as a cell replacement strategy. But on the other hand, this may be one of the lower-lying fruits, one of the sooner rather than later applications of stem cell technology. In fact, there may be clinical trials of this strategy within the next 3 to 5 years.
Let me suggest that this time, if the audience would like to write down questions and pass them to the aisles, ushers will collect them and we'll ask them. Let me go to this side of the table. Dr. Bluestone, you're not a stem cell scientist. You study diabetes and immunology. You just heard the discussion on the brain. Talk about the potential for diabetes.
First, one comment about the brain. I don't do any brain research, but I bet you most of the people in this audience think that if no newcomers come in, how come we're all forgetting and losing our memories as we get older? Anyway, the brain's the toughest nut, I think. It's the one we spend a lot of time talking about, it's the one we think about, but it's a very complex organ with a very complex biology. There are other organs in the body that are actually much simpler. The heart is a pump. And if we could replace the cells in the pump, it could pump faster and better. The pancreas is really a bag of cells called islets whose job it is to make insulin. Every time we have sugar, we make insulin to help make the sugar go away. So I think there are a lot of options in the stem cell field in those organs where we can simply replace much more simplistic functions of the cells. The islets that we work with today in islet transplant, when we take islets from a dead person and replace them in a diabetic, those islets don't go back in the pancreas, they go into the liver. Why? The liver is a very nice environment, they like it, it's easy to survive there and stuff. So the cells we put back in to cure diabetes don't even have to go into the original organ that they came from. So I think that the potential for doing replacement cell therapy, which is what we're talking about here, in diseases like diabetes is great. Now the challenge is, of course, how to make the cells work correctly and how to respond. Why would islets be better than just giving insulin shots 5 times a day? Well, you know, whenever you take an insulin shot, it's like taking aspirin. It works for the first hour great, and then it slowly goes down, your headache comes back, and then you take a lot more. Well, it's the same thing with insulin. It works great when you first inject it, and then it slowly disappears, and you have to take another injection. You go this up and down, up and down. The pancreas is incredibly sensitive. When a little sugar comes, you make a little insulin. When a lot of sugar comes, you make a lot of insulin. So we need to make cells, and I think we can, that will in fact detect blood sugar the same way that your normal islets detected. And then I think you'll be able to control your blood sugar better. So I think in other organs like the liver, like the heart, like the pancreas, we should be able to make cells that have very simple jobs, that don't even necessarily have to live in the precise place they came from, and be able to do the job that they need to do.
What are some of the possible safety issues involved in transplanting stem cells? If you can make these cells into islet cells, what are some of the safety issues involved in transplanting these cells?
Well, there's the first issue about the cells themselves. You've heard a lot about what these cells can do. Well, I certainly wouldn't want to transplant a cell into a liver that's supposed to make insulin and have it start making an eyeball or an ear or a heart. That's not quite what you'd want to do. So you've got to be pretty darn sure that the cell you've created is what we call terminally differentiated, that it's going to make insulin and that's all it's going to make, and it's not going to make anything else.
So you're not transplanting the stem cell?
No, you're actually forcing in a petri dish those pluripotent cells to differentiate, to turn into the ultimate cell you want. And you've got to make sure that the cells you put back into the person are only those cells that differentiated and stopped growing and in fact are making what you want. You don't want them to still have that pluripotent potential after you stick it back into somebody.
Now, how are you going to prove to my former agency that, in fact, all those cells are, in fact, terminally differentiated?
Yeah, I mean, it's a big challenge. It's probably, for my being an end user of this, when these guys give me the cells that are going to work, I need to be sure of that. How am I going to be sure? Well, I'm going to fingerprint the cells, right? I know what a stem cell looks like. I have a genetic fingerprint. There are 30,000 genes that are in every cell in the body, and some of those genes are turned on and some of them are turned off. And I better make sure none of the cells that characterize a stem cell are there anymore. And I better be able to tell in 1 in 100 million that there are no contaminants. And I'm going to have to prove to the agency that there aren't any cells that have that capability. I'll do it by putting those cells into animals first and making sure that they don't grow in animals, that they only make insulin. I'll do it by making sure in some cases that there's a way of getting rid of them. If there's a problem, someone mentioned earlier, you stick the cells in the brain and they don't work, you got to get rid of them if they're not working the way you want. So we're going to need to have, suicide genes so we can go in there and kill off the cells we put in.
Wait a second. Suicide genes?
Suicide genes.
What's a suicide gene?
So every cell in the human body, what's amazing about the body is we spend a lot of time killing our cells as much as we do living them. We were talking about turnover. The average skeleton turns over every 10 years. You have every bone in your body turns over. That doesn't happen without death. When a salamander, when a tadpole turns into a frog, it's got to get rid of that tail. It does it by killing. And so we have to be able to understand how to kill off these cells by taking the natural mechanisms that cells use to die and probably be able to target them if we have to get rid of the cells we put in.
We know how to do this?
We know how to do it experimentally extremely well. We can take a single gene, put it into a cell, and give a drug that would get rid of a cell that's infected with a virus or something and get rid of that cell. We can put that gene into a cell, treat the animal with that drug, ganciclovir, which is an anti-herpes virus drug, and get rid of all those cells. Now, can we get rid of everyone? Well, if I'm the FDA, you better prove to me I can get rid of everyone, because I certainly don't want to, 5 years from now, have a tumor growing from a cell that's pluripotent and can grow into anything you want it to.
This a big issue?
Gigantic.
What do you worry about? What are some of the worries?
I worry about that. I worry about being able to permanently train these cells to do exactly what they originally do. I worry a little bit about going backwards. People talk about going backwards from one cell type to a embryonic stem cell. Well, it took a lot of work for that cell to figure out how to be a fat cell. To be able to go back exactly the right way and then turn it into a beta cell. And, it's like, I don't know, I can't do MapQuest very well, but it's really hard to go backwards and forward. And, the GPS thing never works for me. So I think you got to be real. That worries me, that we're going to try to re-engineer these cells, redifferentiate them, and then get them back where they want to go. And getting all of that right it's a lot of circuits that have to be back in place. That worries me. It worries me that at the end of the day, we'll make these things in a petri dish, we'll stick them into people, and then their immune system, which is very smart and has memory and specificity, will in fact destroy those cells, and we won't figure out good ways to prevent the people we transplant these cells into to keep them and not destroy them over time. And it worries me that in fact we won't have the right technologies to be able to manage these cells once they're in the body to make sure they keep doing what we want them to do.
So let's say for some reason, some unforeseen reason, we really can't transplant these. I mean, it just doesn't work. Are we wasting our money? Anyone?
I can answer that. To me, we would not be wasting our money, and I think that even a lower lying fruit is understanding human embryo development and birth defects. So almost all human embryonic stem cell lines are derived from embryos that were formed in a clinic from eggs and sperm that frequently carry birth defects. I'm an associate professor in obstetrics and gynecology, and I've worked with the clinic a long time. And in generating human embryonic stem cell lines, one of the fundamental things we're learning is what makes a good stem cell line and what makes a bad stem cell line. And by extrapolation, that extrapolates to what makes a good embryo and what makes a bad embryo. And so even that, to me, is really, really very, very worthwhile. And understanding the very early parts of human development, the genes that come on and the genes that go off, that also has been something we've never, ever been able to do before. And so I think that makes it very, very worthwhile.
there are a number of ways to make human embryonic stem cell lines. Teach me.
Well, there's two major ways, I would say. The one is the one I told you about, where you take embryos from an infertility clinic in general and make an embryonic stem cell line by growing the embryo out, and the inner cell mass, spills out into the dish and creates a stem cell line. The other one has gotten quite a bit of press lately, and that's a process called somatic cell nuclear transfer. In somatic cell nuclear transfer, we're taking advantage of a very special property of the egg. The egg is really one of the only cells in the body that can completely reprogram nuclear DNA. So if I look at my skin cell knows it's a skin cell, and when it divides, it's going to form another skin cell. But if I take my skin cell and fuse it to an oocyte that I've taken the nucleus out of, so I take an oocyte or an egg, I take the nucleus out, and I put the nucleus in from my skin cell, the egg is going to completely reprogram that skin cell so that it's now an embryonic cell. It does not identify itself as a skin cell, and it can form any cell type in the body. And recently, in 2004, a group led by Dr. Woo-Suk Wang in South Korea made the first human embryonic stem cell line from nuclear transfer. This is somatic cell nuclear transfer. In that case, they had to use 242 eggs from women to make one line. So that's a lot of eggs by any measure. A year later, they actually had perfected their technique, and some of the things that we read that were perfected are actually very simple. Instead of removing the oocyte if you have an egg and you can put a needle in and pull out the nucleus, instead of doing that, they made a little slit and they gently squeezed and the nucleus pops out. In any way, just those many little changes like that. So it's not really as technologically oriented as some of my colleagues on the right were describing in brain stem cells. With many small technological advances like that, they were able to produce 11 human embryonic stem cell lines that immunologically matched patients, and they, on average, used only 10 eggs for the production of each line. So they went from 242 eggs per line to 10 eggs per line. Okay. Now, the interesting thing about somatic cell nuclear transfer, and I think perhaps Elena could, there's some very problematic issues with it as well. But the interesting thing that it also, to me, really gives to the stem cell field is that the process of reprogramming DNA, a sperm and an egg come together and they make an embryo, and that embryo, all of the genetic information is modified and reprogrammed in that nucleus. We know virtually nothing about that, and the opportunities to learn are just incredible.
So, Dr. Gates, Dr. René Réau-Père just explained to us about somatic cell nuclear transfer. Is that cloning? And what's the difference between therapeutic cloning and reproductive cloning?
I think when we use the term cloning, we tend to think about what Dr. Bishop mentioned with Dolly the sheep, and that was reproductive cloning, where they took a nucleus from a differentiated cell, put it into an egg, and developed it in a dish for the first few days, and then transferred it into a uterus, and it turned into a live infant sheep. We could theoretically do this in humans. If we were to clone you, we could take one of your skin cells, use the process that René described, put the nucleus...
somatic cell nuclear transfer, right?
Into an oocyte.
Take one of my skin cells, put it into an egg.
Grow it in a dish, transfer it into a woman's uterus, go through pregnancy, and your identical clone would be born. Now, universal agreement that we're not going to embark on this
That's
enterprise.
Illegal
In many countries, it is illegal, not everywhere. The ethical consensus around the world is that this is not something that's appropriate to do.
And that's reproductive
That's
cloning.
reproductive cloning.
Now, what's therapeutic cloning?
Therapeutic cloning also, the first steps are the same as what Renee described with transfer of the nucleus into the egg cell. But then, as she described earlier, instead of putting it in uterus, you keep it in a dish, and the outer cell mass dissolves, and the inner cell mass then is available for creation of stem cell lines. And this might get over some of what Dr. Bluestone talked about in terms of rejection of some of these stem cell-derived therapies if one could create a stem cell line that was genetically identical to the person who needed the treatment.
Jeff, why the advantage, I mean, as an immunologist, I mean, let's take liver disease, for example. We can either use human embryonic stem cell lines made from embryos that are sitting in freezers, or we can do what Dr. Gates and Dr. Pera were talking about with regard to somatic cell nuclear transfer. What's the advantages of doing it with regard to this nuclear transfer?
Yeah, I think there's enormous advantages. Everything that makes you you is defined by your nucleus and the genes that get turned on from your nucleus. So anything that's derived from your own nucleus is going to look like you. And so therefore, it's going to be able to be transplanted back in you and have a much lower chance that the immune system is going to see it as different and reject it. There may be some differences. You know, different genes get turned on and off when they're in a petri dish than when they're in your body, and there's probably going to still be some foreignness to the cells, but they're more likely to be much more similar than taking a nucleus from somebody else or taking an embryonic stem cell from somebody else. There are diseases like liver disease and heart disease where clearly is going to be an advantage. In diabetes and some of the other diseases where your immune system has gone awry and seen yourself as foreign anyway and destroyed your own tissue the first time around, that won't be much of an advantage. So in my field, I'm looking for a little bit more than this because the diabetes patients that we see, of course, would reject the new cells just like rejected their first pancreas. But I think for heart and for liver and some other diseases, somatic cell nuclear transplant really has a lot to be said for it.
Is that just a way of making personalized islet cells out of stem cells? So I don't have the issues of rejection, Dr. Crix?
Well, I just want to make sure before we leave this area of somatic cell nuclear transfer that we at least mention one of the other major advantages that this technique offers, and that is the ability to develop disease-specific stem cell lines. So we've discussed using presumably healthy donors like Dean Kessler for these skin cells. But if instead we chose a patient who has, for example, ALS or Parkinson's disease or Alzheimer's disease, and use that nucleus to derive a stem cell line using somatic cell nuclear transfer, then as has been described, those cells will all have the DNA of the patient who has that disease. And if it's a disease of a motor neuron, as ALS is, for example, a neuron in the spinal cord, you could take that embryonic stem cell line and take some of those cells and turn them into spinal motor neurons, that specific kind of cell type in the dish. And those cells would presumably die in the dish the same way they would die in the patient. But you can study that, and you have a human model of a human disease that you can investigate, that you can use for a platform for drug discovery. You can look for conventional therapies that may, drugs that may target the process that kills those cells. And so that whole approach of treating disease that doesn't involve the use of stem cells therapeutically can use the stem cell technology to actually drive that drug discovery process. And that's a very exciting aspect, and it may also be one of the lower-lying fruit in this whole field.
Can I mention one hitch with this somatic cell nuclear transfer, and that is every time we're taking one of these nuclei, we need an egg to transfer it into. Currently, the only place we can get these eggs are from women who donate them through a rather complicated procedure involving ultrasound and long needles that harvest eggs from the ovaries. And that's a concern to a lot of people. What is the source of all of these eggs going to be if we're going to be developing all these different stem cell lines to study these different diseases or create these personalized lines? Now, Dr. Rejo-Pera is working on making oocytes from embryonic stem cells as an ultimate destination for a line, and that might be very helpful.
one other hitch. I mean, these are all, what's exciting about this science is, of course, the challenges as much as the excitement around it. And another hitch is, remember, this is back to the MapQuest, right? This nucleus has already made a lot of decisions. It became a fat cell. And so it's already decided a lot of genes to turn on and a lot of genes to turn off, like the insulin gene. It's not made in fat. So you're asking that nucleus to reformat itself, basically do a hard boot and come back to where it was when it was an embryo. And it may get it right, it may not quite get it right.
So another great question that we're going to be able to answer over the next 5, 10 years, whether these cells are really going to be therapeutically useful?
I think we'll answer a lot of questions in the next 1 to 2 years, maybe 3 years. And one of the first questions is, how efficiently have we rebooted? And that's actually a huge, huge question. So if we put a nucleus into an oocyte as it's maturing, as it's becoming an embryo, when we put that nucleus in actually affects completely the degree of reprogramming that occurs. So that time span actually has to be pretty precisely regulated. So there's some technicalities that no line in the world has ever been shown to be normal by somatic cell nuclear transfer yet. So there's some experiments to do. That's 1 to 2 years. And then should those work, then you could go on to the protocols that Jeff had described.
From Cole Hall, what is the role of stem cells in cancer treatment or cancer etiology? Dr. Bishop.
Well, the answer to that question is an interesting illustration of how important context can be in scientific discovery. We've known for a long time that most cancers originate from a single cell that somehow goes awry and takes a number of steps and eventually becomes malignant. But we've never really known what that cell was in any given tissue. Was it a mature liver cell or was it something else? As the focus in medical biology was turned to stem cells in general and adult stem cells long before embryonic stem cells came into view, came to mind that maybe it's the tissue stem cell, the adult stem cell in a given tissue, that is the vulnerable cell that the causes of cancer act on. After all, this is a cell with what was called renewal capability, the ability to divide over and over again, seemingly indefinitely, infinitely. And that is a property of cancer cells. Cancer cells, we can usually recognize the tissue they're from, but they're not wholly formed. They look like they're still somewhat immature. Well, that brings to mind, too, the precursor cell, something between an embryo, between an adult stem cell and the mature cell. It's now become apparent that in at least several kinds of tumors, there are, in fact, stem cells that are giving rise to the tumor. We call these tumor or cancer stem cells. They are very hard to identify and isolate. We know they're there mainly by inference. We don't know how many kinds of cancers have these yet, but they're there. They're the constant renewing source of the tumor. The tumor cell itself looks a little different, or maybe quite different from the nominal stem cell, but it originated from the tumor stem cell. That's the hypothesis, and there's mounting evidence for this. Now, why is this important? It's important because the therapeutic response of the tumor stem cell could very well be different than the therapeutic response of the mature cancer cell. And until now, when we select therapies, we select them on the basis of the response of the mature cancer cell. If someday we can have the tumor stem cell isolated for study, we will more than likely be isolating therapeutics aimed at the tumor stem cell. I remind you that most tumors, if they're cured at all, malignancies, they're cured by surgery. There are exceptions to this statement, but typically, if a tumor is metastatic, has spread through the body, we can get remissions, but we don't commonly cure such tumors by attack with chemicals and radiation. And suddenly we realize that the reasons for this may not be that we're failing to kill all the mature cancer cells. We're simply failing to eradicate the cancer stem cell. So this is a major transformation in how we think about cancer. It is still probably 3/4 hypothesis, but I'd put good money on it.
So, Dr. Bishop, let me understand. I mean, if I look at a cancer in a tissue, and I look at there are, millions and millions and millions of cells in that.
Billions upon billions, yes.
Right? Of cells in that cancer. And right now, the goal of chemotherapy is to wipe out those cancer cells. And I normally look at those cells and I don't differentiate between one type and the other. I want to kill all those cells. And what you're saying is that
So you'd want to do that, but the point is you've not done enough if this is all correct. You have to also have a therapy that's directed at the cancer stem cell, which may be 1 in a billion.
Just sitting there in that
Yes,
cancer?
right.
And in general, I mean, your work, I mean, cancer arises, I mean, the etiology, I mean...
Well, the etiology, the cause of cancer are external agents by and large, such as cigarette smoke, sunlight, ultraviolet radiation, chemicals. But what they act on are our genes. And cells that are dividing constantly are far more vulnerable to that kind of damage, to permanent genetic damage. And again, that's what the stem cell is doing. That's what the adult stem cell is doing, slowly, constantly dividing. So it is the most vulnerable cell in the body, tissue by tissue, in principle, the most vulnerable cell in the body to external causes of cancer.
So it may not be the mature cell that is in fact becoming the cancer cell.
That's correct. Mature cells in many tissues have stopped dividing completely. And there's a pretty strong school of thought that you can't actually turn a cell that has stopped dividing permanently, that is frozen in place as a mature cell. You can't turn that into a cancer cell because you can't make it go backwards to a dividing cell. That's still hypothesis, but it's certainly in the laboratory very difficult, if not impossible, to make that happen. Whereas the stem cell is dividing, and it is vulnerable to what we call the transformation to the malignant or pre-malignant state. This idea is by far best developed and actually highly credible in leukemias, in blood tumors. And we have the capacity now to identify and to begin to isolate leukemia stem cells, as they're called, to study them and to see whether we're correct that their response to therapeutics is different than the final leukemic cell, and therefore we need 2 stages of therapeutic, the one to wipe out the ultimate cancer cell and the other to wipe out the precursor stem cell.
We're going to get this question sorted out in the next...
I don't make predictions like that.
Another question from Cole Hall. How can you direct a stem cell to mature in a specific direction? Who wants to take it?
Well, this borders on the sort of things that I do in the laboratory because I'm a developmental neuroscientist, and I think the clues to doing that come mostly from studies of normal development, organogenesis or embryogenesis, how in the normally developing animal do these undifferentiated cells turn into a liver cell, a spleen cell, a bone cell, a nerve cell. And by studying the stepwise progression of those lineages, we get clues that will help us do the same thing essentially in a dish, which is what we try to do when we drive a stem cell down a particular pathway. And there are some already examples of what we could call proof of principle, where studying the development in my field of nerve cells in the spinal cord, for example, has given us insights into the kinds of signals that normally drive the fate of those cells. Substances that are diffusible, known as things like retinoic acid and sonic hedgehog, are actually involved in the normal development of these spinal cord cells. With that information in hand, it's been possible to take embryonic stem cells from a mouse blastocyst and treat them the same way, with retinoic acid, with sonic hedgehog, and turn them into spinal cord cells in the dish. And then take those and transplant them back into a developing spinal cord and show that they can integrate and actually send out nerves and innervate the muscles and function like spinal cord nerve cells. So the studies of development are really going to give us the clues we're going to need, I think, to drive this process of differentiation.
A question from here in Genentech Hall. Dr. Gates, how are the ethical issues being dealt with at UCSF?
Before, I think I'll back up a little bit and just say a few things about how ethical issues typically are dealt with in research. And those are through guidelines that are generated nationally, often by the NIH or federal agencies, and come in association with funding of research. These are guidelines having to do with ethical review of research experiments involving humans, informed consent for participation in research. And as many of you know, one of the driving forces behind the California program was the fact that the federal government is not funding research related to embryos and embryonic stem cells. And so we were functioning without these sort of federal guidelines that we typically will get. So here at UCSF, what we did several years ago was form a committee called the Gamete Embryo Stem Cell Research Committee, which is made up of scientists, ethics experts, physicians, some community members, lawyers, and we have just deliberately been working through the various issues as we identify them. We've published a few of our recommendations in the literature, and we're very pleased to see earlier this year that the National Academy of Science came up with some nationally guidelines aimed at scientists around the country, which parallel pretty closely the sorts of ideas that we had been come up with. And that committee continues to work actively here on campus.
Here's a question from our audience at Cole Hall. This one is addressed to Dr. Kriegstein. What exactly is UCSF doing to advance stem cell research in the future?
Well, as all of you know, with the passage of the Proposition 71 initiative in California, there's a unique opportunity here to move forward, especially with human embryonic stem cell research. So at UCSF, we've organized, even prior to Prop 71 passing, a program in developmental and stem cell biology. And I have to admit, I'm very impressed with the notion, and one of the things that attracted me to come here, that the developmental aspects have been linked to the stem cell aspects. I think the basic understanding of developmental biology is really going to be critical. So we have a broad program. It includes developmental biologists, it includes molecular and cellular neurophysiologists and cell biologists. It includes clinicians and the whole breadth of expertise in cancer biology and immunology, and then in each of the organ systems that we think are going to be useful in the future for targeting diseases. The heart has been mentioned already. We have a program in heart attack, the possibility of replacing those heart muscle cells that are injured with new heart muscle cells. The diabetes program, of course, in terms of insulin secretion and islet cell replacement. And the nervous system, we're looking at Parkinson's disease and ALS and spinal cord injury. In the mesenchymal stem cell field, there's a program that's looking at using strategies for stem cell biology to make bone and muscle and cartilage. So we have broadly based programs that involve nearly every aspect of the community here at UCSF. But I should mention that we really depend vitally on support of the community for needs that aren't going to be met by the CIRM. And this falls in two categories. First, in terms of recruitment. So in order to attract the best new scientists we can bring here to the campus and to keep the ones we already have in some cases, requires quite a bit of resources. And those resources are unlikely to come from the CIRM. So in order to develop recruitment packages to bring these people to UCSF will require raising funds through private philanthropy. And the other area is in terms of physical resources, of facilities. As many of you know, in this area of research, the federal government has actually prohibited the use of federal funds for these newer derived human stem cell lines, which we think are some of the most encouraging ones. So to do research with those cells requires private funds and also private facilities. You can't mingle the NIH research with non-NIH funded research. And we think the best way to keep that clearly separate is to do the stem cell research in a dedicated facility. And so we have in mind doing that as well. And that will hopefully involve a partnership with the CIRM, but it'll clearly also involve raising matching funds and involve private philanthropy again. So in order for us to build the world-class program that we have in mind, it's going to be a key component of that is going to have to be a private philanthropy. And I'm very happy that UCSF is actually located, situated in an environment that is friendly toward this kind of research. Not every place in the country can boast that.
Another question from Genentech Hall. Not long ago, there was great excitement about monoclonal antibodies and about gene therapy. But so far, they have not lived up to their potential. Why should we be more optimistic about stem cells? And then the questioner goes on, is taxpaying Californians, will we be greatly disappointed or shown as progressive leaders?
I want to correct the question.
Yeah, I
Nothing
think that
personal, but monoclonal antibodies have transformed drug therapy. There are more monoclonal antibodies that have been approved as drugs in the last 2 years
Gene therapy.
than ever. Oh, well, that's someone else's problem. We're doing okay in the monoclonal antibody.
What did we get wrong in gene therapy?
I think one of the things we've learned about from gene therapy experiments are how to be much more cautious in designing experiments and in involving human subjects in the experiments, because some of our best examples of experiments gone awry are in that specific field.
But you know there is pressure.
Absolutely.
And it's not just the disease advocates. But people will say, people are dying from these diseases. How do you what's the balance here? How do you achieve that balance? You just talked about cautiousness.
I think I've been extremely impressed by the scientists that I'm associated with and how cautious they are. Just the other day we were talking about a possible collaboration with some scientists overseas, and people in them were very cautious that this group might be wanting to move so fast that they wouldn't be deliberate and take into consideration all of the safety concerns that Dr. Bluestone talked about, making sure that we're not transferring things into humans before we know that they're really safe, and being sure that we really engage our participants in understanding what the experiments involve.
Do we really understand what has to be done before we go into clinical trials?
I think one of the things that I heard Jeff say, and I probably haunted the gene therapy field, was not keeping track of the fate of all your cells that are transplanted. So those are genetic, not being confident that these cells will not differentiate into other cell types. And so developing the tools, and there's different ways. If I look at a cell, a very primitive germ cell, I see inside of it which proteins are interacting and how they would fluoresce in certain lights. And there's ways to actually assess the correct structure of your cell that we didn't have even a few years ago. But those types of careful decisions about when is it good enough are actually going to have to be made.
Someone mentioned animal research, too, and maybe one of you who works in animals can talk a little bit more about the value of modeling things in mice and for how long that needs to be go on before we try it in people.
Well, I think that mouse models have really changed the way we do things now. And we can do so many things in mice that we couldn't do before. We can do many things that we used to think we had to do in primates and non-human primates, and we can now do these in mice. And I think that stem cells, when we combine mice with human stem cells, all of a sudden whole new worlds open up to us. And I think, for instance, these kinds of mice that are going to end up with human stem cells, if you think about gene therapy, all of a sudden we're going to have new ways of testing gene therapeutic approaches that we didn't have before. And I think the jury's still out on gene therapy. I think in the years to come we're going to see some dramatic advances. One of the things we were able to do was leap ahead in our vision without all the tools. So we could see where this could go. People got excited about it. People wanted to use it. But the delivery of the genes was really the problem, and we still haven't solved that. This is still a big, big problem.
I think it's worth pointing out that I agree, the gene therapy community promised too much too soon. And there were some very obvious problems to be solved, just as we're talking about obvious problems that need to be solved in stem cell before you could possibly do clinical work. But there are bubble children with full immunological deficiency, who, you remember the bubble child in Texas, who are now living normal lives because of gene therapy. There's been a complication there. Again, there arose from ignorance that will be and can be overcome. But fundamentally, there is an example of where gene therapy has cured an absolutely dreadful disease. So my point is that it's not a lost cause. My point is that too much was promised too soon, and that isn't. I think it's one of the worst mistakes medical scientists can make.
From Genentech Hall, another question. How does the research community respond to the President's restrictions on federal funding for certain types of stem cell research? I want to focus on the science. Talk a little about those stem cell lines.
So there's what are called presidential lines and non-presidential lines, or federal and non-federal. The research community, there's definitely a need for new lines. We're actually hitting some pretty expected barriers to progressing further. And one of those barriers is that the number of vials in the banked low passage cells is rapidly depleting. And the cells are being grown for longer and longer times in vitro, and everyone has always known that's not a good idea. What happens is these cells accumulate genetic mutations. So there are some problems with those cells. On the other hand, we've learned a tremendous amount. And one of the lessons that I think that I've seen in having to concentrate on a few cell lines is we've learned a lot about a few cell lines. And there are 200 or 250 cell lines out there now. But people don't widely use all of those cell lines. One of the restrictions is, one of the problems is that in order to use a non-federal line, a line that can't be supported by federal funds such as the NIH, you have to have the facilities that Arnold was referring to, which we have in obstetrics and gynecology at UCSF, and that's a separate, rented, not incredibly unfancy place where we can do some of the experiments that we need to do. But we certainly would like to integrate a non-federal facility right into the campus community where we could be with our neighbors and actually talk about things.
Let me begin to wrap up. Dr. Carlsson, Dr. Prusner, any concluding thoughts that you may have about tonight's discussion or about stem cells in general, sir?
We are obviously dealing with a very interesting new research field, even though, of course, one can debate how new it is. I mean, still, it's a very dynamic field, and of course, one should pursue many different things here that sound very exciting. But on the other hand, I think one has to be more realistic when it comes to the therapeutic applications, and especially the time it will take before anything. I mean, I think I would guess 2 decades is a too short time here to think that within 2 decades you will have, and at least now I'm talking about the central nervous system, the brain. I doubt very much that within the next 20 years there will be a clinical application to this research field.
Dr. Prusner.
Well, I can only say I hope you're wrong, Arvid.
I hope too. We agree on that. We agree on that. That's for sure.
My hope is that we'll see either stem cells as therapeutics or stem cells as a way to get new drugs, and that this will happen in the next 5 years to 10 years, and that we'll have at least one breakthrough in these degenerative diseases that are so horrible and so resistant to any effective therapy.
Chancellor, closing comments?
Well, even if none of that happens, I think there's no question we'll gain profound understanding into the development of the human organism that will have implications beyond anything any of us could predict. Since this session is connected with the Nobel Foundation and an exhibit about the prizes, I think we should conclude with a thought from Alfred Nobel by way of explaining why he created his prizes, he said he wanted to help dreamers because dreamers have great trouble getting on in life.
That's my problem.
Yes. So what we're talking about here is a dream. This is a dream, and nothing ventured, nothing gained. We really must pursue it to a rigorous conclusion.
Join me in thanking our panelists. There are, we know that there are far more questions that we've been able to answer in this brief time together. Please visit our website, stemcellfacts.ucsf.edu. We'll take many of the questions from tonight's audience and post the answers in the coming weeks.
And there'll be a quiz.
Once again, I would like to thank the consulates of Sweden and Norway. I would also like to recognize our scientific hosts at our telecast sites at the Gladstone Institute, Dr. Rick Deriving, co-director of UCSF's Institute for Stem Cell and Tissue Biology, at Cole Hall, Dr. Susan Fisher, who heads one of the few groups in the country that is developing new human embryonic stem cell lines, and Dr. Michael German, who is studying how to prompt human embryonic stem cells to become pancreatic islet cells, which could be transplanted into patients to treat diabetes. Join me one more time in thanking our panelists. Thank you for all joining us, and good night.